Viewer discretion is advised.
Today we're going to be doing another video of solving difficult questions, but this time we're going to be going over paper number 4. So let's just get right into it. Alright, so describe how a gene can be transferred from one organism to another using genetic modification. Now this question does not look that bad until you consider the marks. So to understand this question fully, obviously you might know the process, but how do you structure your answer for those 6 marks? Let me show you. But before that, let's go over the process. So we're going to be starting off this process with our very own human. Now to understand this completely, you got to think of it in this way. Let's say we want to modify and make insulin for human use. So we're going to be manipulating, we're going to be gaslighting bacteria to produce insulin for us. So how do we do that? Number one, we got to find those insulin genes. And where is insulin made? In the pancreas. So anyways, if you look over here, let's say this is you, and we're going to have the pancreas cells over here. If we zoom into this pancreas, and we're going to see that each of these cells have a nucleus. We zoom into the nucleus and we're going to find the nucleus that contains our chromosomes. Now the chromosomes contain DNA, and let's say this specific section of the DNA is responsible for the production of insulin. So to extract that, we're going to have our scissor enzymes. What are these scissor enzymes called? These are called restriction enzymes. Now restriction enzymes are really important because not only do they separate the codes for insulin, but they're also going to leave behind these sticky ends. Yes, we call them sticky ends. Alright, so what are these sticky ends? These are going to be unpaired bases that we can use for the future to join them up with the bacterial DNA. Moving on, we got Bob. Now we're going to talk about Bob a lot. Bob is my best friend. He's Bob the bacteria. Now why is this bacterium called Bob? Now I remember this because Bob has like a circular shape, and just remember that bacteria starts with a B, has these circular plasmids. I know this is very lame, but it helps a lot, right? So Bob over here is going to have plasmids, which are circular rings of DNA, as you can see over here. Essentially, we're going to use those same restriction enzymes and cut up part of the plasmid DNA. And when we do that, you're going to see that it leaves behind those sticky ends. Now, what were those sticky ends? Again, unpaired bases, do not forget. Now we got to combine these two DNAs, okay? One that codes for the human insulin gene, and the other that is just the normal bacterial plasmid, right? And we do that using our zesty little enzyme, ligase. Get it? Haha, ligase. Okay, guys, the actual spelling is going to be ligase, but like, you get my point. Anyways, ligase over here is going to be joining the sticky ends from both ends, and that's going to form our recombinant plasmid. Recombinant means a new transgenic change in it that resulted in this plasmid over here. Before it used to look like a green circle, now it has a bit of red insulin gene in it. Anyways, we're going to inject this bad boy into Bob, and then Bob is going to have this insulin gene that is infused with one of his plasmids. Bob is going to start reproducing when we place him in a fermenter and form Bobrina and Bobby. Okay, guys, let's just ignore this. The main point over here is we're going to ferment the bacteria so that it can produce more copies of itself and that we can have greater production of insulin. Now, how are we going to structure our answer for those 6 marks? Let me show you. Over here, you can see that the ones that I highlight yellow are going to be the keywords that are the ones that are giving you the marks. So the marks over here are from number 1, isolating the organism's DNA that we saw. Like for example, we talked about the pancreatic cells. We use restriction enzymes, and the very important word, and you cannot miss out on this word, that is going to be the sticky ends. Okay, after we have that, the bacterial plasmid DNA is also cut, and similarly we're going to use the same restriction enzyme. Same is very important. And this again, what does it form? Remember those sticky ends? The next process is the sticky ends of the organism's DNA and the plasmid DNA are complementary. So this is very important. The organism's DNA is inserted into the plasmid DNA using the gay enzyme that we talked about, the ligase enzyme. And this results in the formation of, another keyword, recombinant plasmid. The recombinant plasmid is inserted back into the bacterium, right, as we mentioned, and then a fermenter is used to. Now this is very good. This is a very good word. Examiners love maximize. Over here, you got to see that these keywords are linked together using filler words, and these filler words are important because that's what makes your answer flow in a sequence. Like for example, we have the word similarly. We have words like forming, leaving. We have important uses of the full stops, the commas, right? So these things really matter when it comes to making your answer flow like a river. Okay, next up we have describe differences and similarities between the structure of a plant root hair cell and a bacterial cell. Now this might look easy on the surface level, but if we scroll back down again, 5 marker, here we have our trusty root hair cell that's gonna be competing with Bob. Now Bob has evolved over here. You can clearly see that he is looking pretty sharp. So simply put, we have the nucleus, ribosomes, mitochondria, cytoplasm, cell wall, and cell membrane. Now if you talk about Bob, Bob has also some certain features. Let's look at them. So over here, we're going to have ribosomes, cell wall, cell membrane, plasmid, and free circular DNA. If you guys recall, we use the plasmids for genetic modification. So over here, we're going to see a few differences. As we mentioned, the root hair cells have a nucleus, they have a mitochondria, vacuole, cellulose cell wall, they don't have plasmids, and they have linear DNA. Whereas the bacteria used to have the circular DNA. And over here, root hair cells and bacteria, they both have cell wall. We talked about this, even though the composition is different, but the structure is still the same. They also have cell membrane, cytoplasm, DNA, and ribosomes. What happened here? So in accordance with the marking scheme, you just got to give a maximum of 3. So if you have 3, then you're good to go. And similarly over here, 2 to 3 should be fine as well. So 3 maximum over here too. Okay, so next up we have a pretty long question. Let's go over this real quick.
Scientists investigate the effect of changes in pH. We're measuring pH and the activity of 2 different enzymes. So each enzyme is placed in a hole in the center of a jelly layer in a petri dish. The jelly layer was made of protein. Now that makes sense because we're using protein enzymes that have to digest protein, so you can see that something is happening. Over here, the investigation was carried out at a range of different pH values. After 60 minutes, the area of the jelly layer that had been broken down by enzyme L or M was recorded at each pH value tested. So this is like a demonstration over here. Here we have the protein. Here we have the protein jelly layer. The hole in the middle is going to be a solution of either L or M enzyme. Okay, and here we have the investigation results.
Now, why did I choose this question? Because we just need to describe the graph. The thing is, most students don't really know how to describe graphs, so this is why I've selected this question. So in my opinion, the best way to go about with this question is number 1, to describe what's happening in the beginning with both the variables over here, right? And then we're going to describe with the course of these variables how they change as you move along the ranges of pH. And then we're going to compare these two enzymes towards the end and talk about what's the main differences between enzyme L and M. And this is how you describe results. For describing results, you don't need to explain anything. It's really simple. You just state what you're seeing. Please don't overcomplicate this, please. Now this answer over here essentially summarizes all the findings. So let's go over it again. Number 1, we first said that we're going to start from the beginning. So at pH 1, enzyme L has digested 80 mm² of the jelly layer. Keep in mind when you're quoting data, please include the units. Over here, the jelly layer compared to enzyme M that has 0, right? So from pH 1 to 2, the enzyme L now rises to 160. And from pH 2 to 5, it falls back down to 0 area of jelly digested. The enzyme L remains at 0 for the following pH. So if you go back up, we can see that our data matches the activity of enzyme L. Now moving on, enzyme M starts to rise from pH 3 to 7, reaching 80 mm² of area of jelly broken down, right? So the area remains constant from pH 7 to 8. And if you go back up, we can see this exact trend over here. It rises from pH 3 and then it becomes constant, and then it's going to rise again and fall. So let's talk about that. So over here, it rises again from pH 8 to 10, reaching its optimum. Now the word optimum is very key. Jelly breakdown of 140. Now at the end, if you have space available, please make like a comparison statements, like this is describing the overall results. So we're going to say that the optimum pH of L is at 2, and for M is pH 10. Okay, moving on, figure 2.1 shows the structure of the eye. It also shows the pathway taken by nerve impulses. So draw a label line and a letter X on the figure to identify a motor neuron. We're going to have our sense organ over here, the eye, that is going to act as a receptor. Now the sensory neurons are actually going to pick up this stimuli and deliver it to the CNS, central nervous system, which is the brain over here. The brain is going to relay this information through the relay neuron into the motor neuron, and the motor neuron is responsible for output. So we would see that the neuron that is actually heading back into the eye, that should be the motor neuron. So over here we can see that this portion over here, this is going to be the motor neuron since it's heading back into the eye. Now alongside this question, I found this part, which is describe the distribution and function of rods and cones. Now this is a very weird question because when did we study this, you know? The hell is a rod and a cone? So essentially what you're going to know is rods and cones are going to be the receptor cells, okay? And these receptor cells are located in the retina portion. So if I show you guys this diagram over here, you're going to see that the retina actually consists of the rods and cones, and you got to know that rods is going to be for light intensity, And the cone, since it starts with C, is for color, detecting color. You just got to know that both the rods and the cones are found throughout the retina. However, there's a point called fovea. Now, fovea is going to be that triangular like bulge from the retina that is actually going to contain most of the cones. And then we're going to have the blind spot, which is going to be the eye. So if I show you guys over here, so over here we can clearly see that the rods and cones are going to be located along the retina. They're not going to be present here because this is the optic nerve. This is the blind spot. And there's going to be a distinct point where all the cones are concentrated. That is known as the fovea. Let me see if I can see it. I think this is the fovea. So over here, we're going to see that the answer is going to be that both of the rods and the cones are located in the retina, and there's going to be higher concentrations of cones in the fovea, whereas no rods, no cones are in the blind spot. These are all keywords. You got to mention this or you're not getting the mark. You can't just write there's no rods and cones at the invisible spot. Please write down blind spot. Now, rods are mainly concerned with light, so they're going to be like light receptors over here, and they're also going to detect low light intensities. So essentially, rods, whatever light you perceive, they also convert it into electrical stimuli. I could have mentioned this here, but I just kept it brief. Don't overcomplicate things, Cones are going to detect the opposite bright color, brighter, increasing light intensity, and also detects color. Remember, C for cones, C for color. Here we have another interesting question. Describe the pathway taken by the products of digestion, protein digestion, from the villi to the liver. You know what these small tiny things are called? These are called villi, and they're so like you just want to like swish them, like they're super satisfying to look at. Anyways, if you zoom into this little guy over here, this is not Bob, don't worry. We're gonna have this villi, and this is what the structure of a villus looks like. Now over here, you gotta keep some things in mind. I'm not gonna go into detail, but you gotta know that the blood supply is like really dense in the villi, and the venous system is gonna be draining the products of, let's say, protein digestion, which is gonna be amino acids. They're gonna pass single cell layer, and they're gonna enter through diffusion, and they're going to start moving over here, and the amino acids are essentially going to head into the liver using the hepatic portal vein. Now, this is very important. This is something that most students don't know. They think that the vein that is actually responsible for bringing the gut blood into the liver is the hepatic vein, but no, it's going to be the hepatic portal vein. Okay, so don't mess this up. So essentially, this should be our answer. We're going to say that the amino acids are going to move into the capillaries by the process of diffusion, and then these guys are going to be carried by the blood plasma towards the venous system that we mentioned over here. Very important. Please, without this, your question is invalid. Hepatic portal vein that supplies it to the liver. Via means through. It's like a really fancy word. Thank you. Now, this is a very interesting question. So over here, we had a tree that essentially got cut from this section, right? And we need to explain why the area above the girdle will become swollen. So essentially this area over here, why does this area become swollen compared to the one below? Now, to understand this, you got to have a good idea of translocation and how it works when we're moving food, sucrose from source to the sink. So you already know your boy got those diagrams ready and set. This is going to be our old wise mystic tree. And we can see over here, if we cut a section of the bark of the stem of the tree, we're going to see a pattern like this, where the xylems face inwards and the phloems face outwards. So if you think about it, if we're cutting the bark from right over here, we're essentially going to be removing the phloem section, which is responsible for what? Movement of food. Now again, let's say we evaporate these poor phloems. Oh, not my drawing, man. So this is what's going to be left behind, just a bunch of sad xylems. So over here, you can see that clearly, if we have photosynthesizing leaves are going to produce sucrose, this sucrose is going to get loaded into the phloem, and the phloem will try to go down through the phloem tubules. But however, they're going to get blocked over here because we cut down the phloem, and that is why the sugars begin to accumulate over here, causing it to bulge, swell. Hopefully that is clear. Now with the snap of my fingers, the answer is going to appear in 3, 2, 1. Okay, so over here we can see that the answer is going to consist of number 1. We're going to have photosynthesizing leaves that produce glucose, obviously, because carbon dioxide in the water, they mix, they form glucose and oxygen. And then this glucose is actually converted into sucrose. Now you can think of it like this. Glucose is like the simple sugar, okay? We need to convert it into a form that can be transported. That is sucrose. Okay. Now sucrose is translocated. Very important word, translocation. Please don't miss this out. Translocated from the leaves, and again, very important, we mentioned the main thing the question was talking about, source and sink. This is going to be the source, And we're going to have the roots, which act as a sink. And obviously, this is going to be flowing through the phloem vessels. Now, the phloem transports the sucrose and is going to get blocked at the level of the girdle, because why? The phloems are cut, so it cannot really move. This leads to the accumulation of sucrose, causing the area to swell. So these were some of the hardest questions that I could find from the years, I guess, 2023 to 2024. So if you guys have any questions, do let me know. Yeah, I'll see you guys next time.